Epigenetics studies heritable changes in gene expression that don't alter the DNA sequence, with DNA methylation being one of the most important mechanisms. Methylation can turn genes on or off, and abnormal patterns have been linked to aging, cancers, autoimmune diseases, and neurological disorders like Alzheimer's. Studies have shown that DNA methylation patterns can even predict biological age with a median error of just 3 to 4 years. In Alzheimer's, certain regions like the entorhinal cortex show decreased methylation, while genes like HOXP6 are hypermethylated in patient blood samples, highlighting potential biomarkers for disease. The objective of our study was to investigate the relationship between DNA methylation, aging, and Alzheimer's risk. We developed two predictive models, one to estimate chronological age and another to classify Alzheimer's status from methylation profiles. We also analyzed which CPG sites contributed most to these models and identified the genes linked to the top 20 sites to better understand their biological relevance. We analyzed 1,026 samples from 247 Alzheimer's patients, 203 mild cognitive impairment patients, and 685 controls, which 43% of them were male. Data came from Illumina 850 and 450 methylation B-chip arrays, covering around 894,000 CPG sites, which 510,000 missing values, which measure hundreds of thousands of CPG sites per sample at a fraction of the cost and time of Sanger sequencing. Unlike Sanger, which sequences one DNA fragment at a time and requires bisulfate conversion, Illumina's massively parallel sequencing processes millions of DNA molecules simultaneously, enabling fast, large-scale analysis of our cohort. On the front end, we built a Next.js app with React using shadcn/UI and Radix UI for accessible components. Users can upload CSV methylation data, which is sent to our fast API backend. The backend runs our CNN and XGBoost models and returns predictions. This is loading.
Not here.
This process usually takes 2 to 5 minutes, depending on the size of the data. We then display results in an interactive dashboard showing risk distributions, feature importance, model metrics, and individual sample analyses. Users can also download or share reports. For data exploration, we applied PCC and UMAP visualizations. The PCC plot revealed 3 distinct feature clusters with clear separation of control, Alzheimer's and MCI samples, while UMAP showed meaningful groupings despite some overlap likely caused by noise. Both visualizations support a real relationship between methylation patterns and cognitive disease. In short, our project demonstrates that blood methylation profiles can provide insights into aging and Alzheimer's risk, offering a path towards early detection, personalized insights, and improved understanding of neurodegeneration. Thank you.